Thank you so much. So I'm really excited to be here. I've been doing a lot of talks mostly for biologists, so this is kind of nice to actually be able to talk to some social scientists as well. So I started my kind of research career as a demographer and then slowly transitioned to more of a biologist, but I really like kind of coming back to my roots. So I kind of like to show this picture of tree rings. So this is what most of you probably know how we would age a tree. So kind of what my work is can we actually find measures akin to this for humans or different animal models? Although in this case, it's probably tracking chronological age and weather changes, but can we actually measure something that's tracking biological ages in a human? So the reason that I am very interested in aging is that it happens to be one of the biggest risk factors for most of the chronic diseases that people suffer from. So in any regression model, if you're going to predict risk for developing cancer, Alzheimer's disease, cardiovascular, you control for age. It's the number one thing that's going to contribute. And the reason that we think this is happening is there's actually what we consider hallmarks of aging that are changing. So some of them are genomic instability, telomere attrition, which some of you might have heard of, loss of proteostasis, cellular senescence. One that I study a lot of and that I'll talk about today are epigenetic alterations. And that we think that these really accumulate over time are actually drivers in the pathogenesis of developing these different chronic diseases. So the idea is instead of studying diseases individually one by one, if we could actually identify this fundamental process that's contributing to all of them, we would hopefully have a bigger impact in prevention or elimination of these different diseases. So I like to take kind of what I consider a systems biology approach to studying aging. So this idea that we have different networks and changes that happen at different what we consider biological levels. So you might start with changes at the genetic level that then impact changes at the molecular level, the cellular level, the organ level, and ultimately this contributes to kind of these manifestations of aging that we see in the whole organism. And so the idea is that you can we're set up to be able to tolerate some level of damage at each kind of level before you really see this manifest up to the higher levels. And this really drives some of the thinking. So this is based on a paper that I did with Luigi Ferrucci, who's the scientific director at the NIA. And the idea is that you have kind of these age-related changes at these different levels. So you have biological aging, which we consider as more of these molecular changes. And then, this will start to decline much earlier in the life course. And over time, this will reach some critical threshold where then you'll start seeing these changes in phenotypic aging, so physical functioning, and things like blood pressure, different metabolic markers. And over time, this will then lead to declines in what we see as functional age, so frailty, physical disability. And really, the idea is that you could actually measure aging at any one of these and probably be able to actually detect the declines much earlier if we're actually measuring this at the biological level. So even though we think of these different hallmarks of aging or changes that are synonymous with aging, there's a lot of heterogeneity among individuals in terms of both their aging rates and the manner in which they age. So we think that chronological age is actually a very imperfect proxy for this latent concept we're trying to capture of biological aging that's actually leading to increased risk of different chronic diseases. So the goal of my lab and a number of other labs is can we actually quantify biological age in contrast to chronological age. And the idea is that this will actually give us a much better predictor of risk for developing disease. It also can provide some sort of endophenotype for figuring out what's contributing to differences in aging. It'll facilitate evaluation of interventions aimed at delaying aging, because in humans, we think if we intervene in aging much earlier in the life course, that's going to have the biggest impact, but you then don't want to run a 60-year trial to see who's going to die. So if you can actually have kind of these proxy measures, you could run your intervention, check 1 year later, and actually hopefully see changes in these different aging measures. And then another important thing is it actually can identify at-risk individuals potentially much earlier in this process. And then you can either try and develop interventions for them. So the idea of biological age is if you imagine that you have a group of people who are all maybe chronologically 40 years old, the idea is that we can give them each a biological age, or what I'll mention at the very end, we actually think that they probably should have multiple ages, and that this should differentiate their risk of disease. So this woman should have a lower risk of developing any of the different chronic diseases compared to this man because she has a younger biological age, even though they're chronologically the same age. So in trying to develop these different biological age measures, kind of what is the goal? What makes a good aging biomarker? So number one, should be minimally invasive. Obviously you can't get brain biopsies from people and say, oh, we're going to predict your risk of Alzheimer's disease. So we want to do things you can actually do in the field and get enough people that'll consent to them. Number two, it should track chronological age. So that's obvious. It should change or increase with age. Number three that I think is often overlooked, so I get asked to review papers on new biomarkers of aging all the time, and I hardly ever see people demonstrating this. They show that it tracks chronological age, but they don't actually show if it's a better predictor of aging outcomes than chronological age. But like I said before, chronological age is not a perfect proxy of this biological aging process, and we're actually trying to capture something that's a better indicator of that. So this is the way that you would actually be able to validate it. So if it predicts something like mortality better than chronological age itself. Hopefully it's responsive to intervention or to lifestyles, because what's the point of knowing your biological age if you can't do anything about it? That's kind of how I feel about it. And then I also hope, coming from a biological perspective, that it'll also kind of facilitate some of our understanding of the basic biology of aging. So why do we see these changes with time? So I started this work in my doctoral program at USC. When I did my first paper on creating one of these biological age measures. I'm not going to talk too much about that, but basically what I did was you can take data from 10 different clinical biomarkers, and you can create one of these biological age measures and show that it actually is a better predictor of something like all-cause mortality compared to chronological age. And then this measure was actually used again by Dan Belsky. He had data from the Dunedin cohort, where there are individuals who are all the same chronological age. And again, he could show that there's some variance in terms of their biological ages, and that maps on to differences in things like physical functioning, cognitive functioning, and even, interestingly, facial aging was correlated with this biological age measure. But today I'm going to talk about a new measure that we have that's actually slightly better than what I consider biological age, maybe 1.0, so maybe this is 2.0. So we call this one phenotypic age, because again, we're using these clinical chemistry measures, so things like glucose levels, kidney liver panels, cell counts. So again, it's not at the molecular level, it's more at the physiological level. So for this, what we had is data from the Haynes cohort, which is considered a nationally representative sample. Of course, it's not perfectly nationally representative when you actually get just the lab-based sample. But we had about 10,000 individuals, ages 20 and above, and we have 23 years of mortality follow-up. And so what we can actually do is we use what are called supervised machine learning approaches, so we can actually train a predictor of our outcome, which in this case is mortality from major age-related diseases, so things like diabetes, cancer, cardiovascular disease, Alzheimer's disease, and lung. And we start with 42 of the clinical biomarkers that are in that data set, and we basically train a predictor of mortality. And it ends up just being this linear measure, so we select out these 9 biomarkers plus age, which end up going into this measure, which is just basically a weighted sum of these measures. And then we can actually convert this to units of years by mapping this onto a Gompertz. So basically, it equates, so your measure is associated with some mortality risk, and then we look in the general population, the age associated with that mortality risk, and that's the biological age that we give the individual. based on this Gompertz mortality. Yes.
So I realize that you used the training algorithm to come up with those specific markers.
Yes.
So you can't tell us why you chose them.
So yeah, I did not choose them.
Right.
The
But
computer chose them.
In retrospect, just
Yeah.
looking at it from my perspective, red cell distribution width is a very funny marker.
We don't know why it ends
Why
up
does
in
that
here.
come up?
I don't know. We are not the first ones to so people have showed mortality associations with that measure. We don't know what it's capturing. The way I think of it is that because it's a distribution of the width, that is the more variance you have, maybe in your red cell width, that might be a marker of something going.
Efficiency is a typical.
Yeah. So it's not just that you have increased width or decreased, but you just have more variance. That might be, with a lot of things we see increased variance with age.
RDW
but-
is used clinically.
Yeah.
To look for iron deficiency, hemoglobinopathies. Maybe that's what.
Yeah. So I'm not sure why. Interestingly, I think 6 out of 9 of these measures were in our original biological age measure, which I actually went in and selected measures as long as they correlated with age based on wanting to cover multi-systems. So it was actually really nice to find that the machine learning one actually picked out the same ones I did when I just did a more kind of a priori selection of biomarkers. Yes.
Just a clarifying question. What was your analysis time? Is it age or time, like calendar time?
So for this one, it was calendar time because age ends up, it's in the model. So yeah, then it's just time from blood collection to mortality or censoring. So then we have, the nice thing about NHANES is that they have multiple waves, but they're supposedly not the same individuals. So then we can use the NHANES 4 as our validation sample. For this, we have, I think it was about 18 years of mortality follow-up. And we compared, this is after adjustment for chronological age, because our measure is highly confounded by chronological age. We find that a 1-year increase in your phenotypic age relative to your chronological age is associated with a 9% increase in your risk of all-cause mortality. And similarly, we find when we do cause-specific mortality in a competing risk type models. We also know the number of coexisting diseases a person's been diagnosed with. This is at baseline, so this is not doing any sort of longitudinal, but at baseline, individuals who have more coexisting conditions tended to have higher what we consider phenotypic ages relative to their chronological ages, and actually seems to have almost like a dose response increase in that. So then the other thing is, so this is an ROC curve where we can compare how accurate our predictor is at predicting something like, for this was 10-year incidence of all-cause mortality. And we're comparing it to a lot of the measures that are typically used for doing different risk assessments by physicians. So ours is in red, so the higher you are on this, the higher your area under the curve is, the more accurate your predictor is considered to be. So what we find is, even though it doesn't look that different, even with a Bonferroni correction for all these tests, we find that ours is significantly better at predicting all-cause mortality than age. I was actually shocked at how low disease count is on this AUC, and then something like BMI is not even better than chance, essentially, in this model. And I didn't mention this is an adjusted model for education, race, ethnicity, and sex, and age, except not age, because age is in there separately. So the other thing we did is, to really figure out, maybe we're just getting an end-of-life phenotype. So what it's doing is picking up the sickest of the sick who are about to die. So what we tried to do is go back, or maybe it's a metabolic syndrome or a BMI measure. So we actually did a ton of different stratified analyses to see how it's predicting within different subpopulations. So we did this by race ethnicity first, so this is self-reported. What you can see here is that it's essentially doing just as well in terms of its all-cause mortality association, regardless of which group you look at. Same thing, no difference when we look by education, no difference when we look by smoking, binge drinking, you get slightly higher for people who are binge drinkers, but the non-binge drinkers look, it does just as well. Same thing, disease count, which I was really surprised. I was thinking that among sick people, it actually might be more predictive, but it doesn't seem to. BMI, so underweight individuals is the one place where we really kind of see an improvement. And what I think that is that there's different reasons people are underweight. So some people might naturally be underweight. Some people might be underweight because they're sick. So I actually think that's the one place where it really is differentiating these types of people. And then the one I was most excited about was what we consider this healthy group. So these are people with no diseases at baseline and normal BMIs and don't smoke or have other health behaviors. And it seems to predict mortality even among these people who we think are seemingly healthy. And by looking at them, you shouldn't be able to differentiate their mortality risk. So the next question is, what's contributing to these differences in this phenotypic age measure among people who are the same chronological age? So for this, we really tried to take a life course perspective. We were kind of limited by the data. But we used the Health and Retirement Study, which is a study of individuals ages 52 and above. Supposedly, it's supposed to be nationally representative, but because we were limited to people who did this, what's considered the leave behind portion, and this life history questionnaire, we're not assuming our sample is not nationally representative for this. We did use sample weights, but I'm not going to say anything about representativeness. So we had about 2,000 people who had data for both all the biomarkers and all of the variables we considered. And we used a number of different modeling techniques to try and kind of parse out what's contributing to these differences. So one of them is the Shapley value decomposition method, which is estimating the overall and relative importance of what we consider these different domains in contributing to our outcome. And then we also use a hierarchical, that's misspelled, should be hierarchical clustering method, where we're actually grouping individuals who seem to have the same characteristics, and then looking at these different kind of what we consider subpopulations based on this clustering method. So that's redundant. So for the Shapley value decomposition method, so this is mainly done in collaboration with Shi Chen, who's at Yale, who's an economist who really specializes in this method. So the different variables that we considered in this, HRS provides a bunch of these polygenic scores. So basically, your risk based on genetics associated or for a bunch of different outcomes. So we had 4 different anthropometric outcomes, so things like height, BMI, et cetera. We had 4 different disease longevity outcomes. I think it was parental lifespan. I know heart disease, I forget exactly which ones were in there. 11 of these mental health personality ones, 2 for education and or cognition, and then 2 for smoking. So then we also had information on childhood SES and child adversity and stress, adult SES, adult adversity and stress, and then behaviors and also demographics. In this case, it was just sex and race ethnicity. So what you can do with this Shapley value decomposition is you put these all into the model, and it can estimate their relative importance in contributing to this phenotypic age measure. And this is what we're looking at here is whether you're how much higher or lower you are than your chronological age. So these are our results. So basically what we find is that 9.2 of this, it comes down to these behavioral differences. So things, for this we had smoking, alcohol consumption, exercise. We put BMI in there as a proxy for nutrition, even though BMI is not a behavior. We also get a lot in terms of this adult adversity variable, so almost equal to BMI. Slightly smaller for, here, where's the childhood one? So here, childhood SES, in this case, only 0.7% of the variance is explained by that, and childhood adversity slightly more, 2.1. And then the genetics altogether were, so this is demographics, so here's all the genetic variables. So actually, I was surprised how much of the variance was explained by these different genetics. And we did run this stratified by race, ethnicity, and in this result, we do actually find really similar proportion of the variance explained in both non-Hispanic whites, Hispanics, and non-Hispanic blacks. Or actually, we didn't have the PGS for Hispanics, so it was just non-Hispanic whites and non-Hispanic blacks. So next, we try to do this hierarchical clustering. So this idea is if we looked at people across all these different domains and variables, can we kind of put them into groups of people who seem to share similar characteristics? For this, we didn't group them with the behaviors, all based on things like SES, adversity, and environmental variables. So this is what we call a dendrogram. So if you think of this, every little line going down is an individual, and you would cut this at some height, and if you're connected to a different line below that height, you get placed into a group. And we color code these groups so that we can keep track of them. So we find 6 different, what we consider clusters or modules. I'm sorry, this is really hard to see. But essentially, then what we can do is we can look at kind of what are the characteristics of people in these different modules. And I'll read this off because I know it's probably really hard to see. So this red module tends to be people with high neighborhood disorder and also low education and low wealth. We reverse coded education and wealth in this case. The yellow module are people who tend to have high chronic stressors and somewhat high life events, and to some extent low education and wealth, but not as much maybe as this red group. The green is the almost polar opposite of the red, and then this blue is the opposite of this yellow, and then orange is people who have, this is more of kind of the childhood variables, so people have higher childhood traumas and self-reported family poverty. So now what we can do is compare the phenotypic ages of people in these different groups. So basically what we find is that individuals in this green group tend to have the lowest phenotypic ages. So these are people high SES, so high education, high wealth, and low neighborhood physical disorder, compared to people in this red group, which is the high neighborhood physical disorder, low education, low wealth. And same thing, people in the yellow, which is high chronic stressors, high life events variables. So they tend to have accelerated, what we consider accelerated phenotypic age. I actually left out. There's one more slide on here that I didn't put. So we actually did G by E. So we took one of the polygenic scores that explained most of the variance in our phenotypic age measure. So this was the coronary heart disease polygenic score. And we actually find a really strong G by E interaction in that case. So people who have higher scores on this polygenic score and are in that red group versus the green group have kind of a multiplicative increase in their phenotypic age. However, we were finding some weird differences by race ethnicity to where this is actually amplified in people of African ancestry based on the SNP data. And so we're still kind of trying to tease that out because it's actually the opposite of what we'd expect since most of these polygenic scores are designed for European ancestry. But, okay, so the next question is, so that variable is really showing you it's operating kind of at this level, this phenotypic age variable. But can we actually identify changes that are happening at kind of even a lower level of biological organization that are probably preceding this phenotypic age? So hopefully then you can actually identify things earlier in the life course and potentially get closer to kind of mechanism. So for this, some of you may be familiar with these things called epigenetic clocks, but if not, I'm going to give kind of a very brief overview of what I mean by this. So here is double-stranded DNA, and you have these different nucleotides, so adenine, guanine, thymine, cytosine, which here are represented by these different colors. So the idea is, so if you have, if you represent each one, so those of you familiar with SNPs, you can not pay attention because it's very basic. If you represent each one by the first letter, so you have T, C, C, G. And so basically what we do is if you look across one of these strands, so from what we consider the 5' end to the 3' end, you have these areas where you have a cytosine followed by a guanine. These are called CpG nucleotides, or for short, just CpGs. So these CpG sites can become methylated. So you have attachment of this methyl group. And usually what this does, depending on where it is in the genome, is it can change gene transcription. Or at least that's how we think about it. But the really interesting thing is you actually get really strong patterning in these changes in DNA methylation with chronological age. So maybe if I was looking at an individual who is 0, maybe they would have no methylation here, none, and none. This is just a very abstract kind of example of this. But if I was looking at a 10-year-old, maybe they would have methylation here, none, and none. Or a 50-year-old, maybe they'd be methylated in all 4 regions. So you actually get at very precise points, these really strong age correlations in terms of these changes in methylation patterns. And because of that, people have developed what we call these epigenetic clocks. So essentially, these are age predictors based on genome-wide DNA methylation. So in the case of what's considered the Horvath clock, he has basically 353 different CpGs so across the entire genome. And by just knowing your methylation pattern, he creates kind of this weighted score that produces what we consider your predicted age, just based solely on your methylation. And what you can see is this is extremely highly correlated with your chronological age. So this is a really good age predictor. So knowing nothing else about you, if I know your methylation pattern, I can predict how old you are. So this is an example of a different clock by Hannum et al. This has 71 CpGs, not the same CpGs as the Horvath clock. And again, you get this really strong pattern. Some of you might notice this part. Steve Horvath actually adjusted for this. You actually get this exponential increase before development that was adjusted for in this, but still in adults, it seems to be a very good age predictor. But what we argue is, so the way these clocks are developed, again, are using these what we consider supervised machine learning approaches. So your computer is trying to find the best predictor of your chronological age. So the way, if you think about it, is this, any deviant or deviation from your chronological age, according to this measure, is treated as error. But what we actually think, we want this variance across chronological age. So can we actually build in something to find what we might consider the true residual? So it's not error, it's not a problem with the prediction, but we actually want to build, this is what we're trying to capture. So the way we're going to do that is we actually use the phenotypic age measure that I showed you in the beginning and substitute that for the chronological age. So we developed a new clock, which instead of training a predictor of chronological age, we're now training a predictor of this phenotypic age measure. And again, I showed you that we validated its associations with all these different outcomes. Then we're going to do the phenotypic age. In this paper, we validated with all these outcomes. I don't have time to show all of them in this case. But we end up with 513 CpGs in this new clock. So for this, we use data from Inchianti. This is an Italian population. We only have 456 individuals. And we start with DNA methylation in whole blood. So we start with about 20,000 input variables that we're going to predict this phenotypic age measure. And we use a model called an elastic net, which again, it just ends up being a weighted sum of methylation at 513 different CpG sites. And what we find is that after adjusting out chronological age, we get a hazard ratio for all-cause mortality of 1.05. So 5% increase in your risk of all-cause mortality for every 1 year increase in your epigenetic age relative to your chronological age, essentially. And here's one of those ROC curves again, where we're comparing our mortality prediction with that of the Hannum and the Horvath measures that I originally showed you. And we find that this one produces a much better mortality prediction using, in this case, we actually had 9 different independent data sets that we validated this in. So aside from all-cause mortality, we also find that it's associated with things like breast cancer incidence. So women who are breast cancer free at baseline, having a higher epigenetic age is associated with higher risk of developing breast cancer. Same thing for lung cancer, particularly if you're a current smoker. Coronary heart disease. We find that individuals who have centenarian parents have younger epigenetic ages on average. I think Down syndrome was associated with a 12-year increase in your epigenetic age. And then we also find it for things like physical functioning, HIV infection. This is supposed to be mild cognitive impairment. And again, that multimorbidity number I showed you in the beginning, so the more diseases you have at baseline. Yes.
Well, what age did you measure the Down syndrome children? That's a really fascinating observation.
I actually don't remember offhand, but I can go back and look at what that was.
Because these children, young adults have a virtually universal incidence of premature Alzheimer's.
Yeah, so we treated the Down syndrome almost like a progeroid kind of syndrome in this case. They weren't, I'm going to guess they were in their 20s, just based off memory, but I can double check that for you. But yeah, I think, that was one of the interesting things. And so everything I showed you so far is in whole blood. The exciting thing about these methylation measures versus the phenotypic ones I showed you before, is you can actually get an epigenetic age for any tissue or cell type in your body. So I can measure the age of your skin, the age of your heart, if I actually had access to it, the age of your inside of your cheek, anything. But the question is whether, since we train this in blood, whether it actually works in other tissues. So essentially what we find is, yes, it does. We had 32 different tissues or cell types. This is them all kind of graphed on top of each other with different colors, denoting the different types. I don't have it labeled here, but the interesting thing is you can kind of see there's this group versus this group. So all the ones on the bottom are actually from brain. So what we find is actually brain seems to have both a lower intercept and a lower slope in terms of epigenetic aging, which is something we're following up on. But it still has a really good age correlation, so about 0.83. Saliva is something we're excited about because remember when I mentioned minimally invasive, this is a really good sample to be able to get from people. And I know Fragile Families does methylation in saliva. And then also when we look at different blood cell types, we get good age correlations. And then the question is, okay, it tracks with age, but does it predict things within that tissue? So we had data from DNA methylation in brain, so in dorsolateral prefrontal cortex. So we can predict someone's epigenetic age in their brain. These are all postmortem samples, as you'd expect. And we have full neuropathology workups on these individuals. And what we find is that people who are diagnosed with Alzheimer's disease postmortem have higher epigenetic age in the brain. And these are different hallmarks of Alzheimer's disease, so amyloid and neurofibrillary tangles and plaques. Same thing when we look at idiopathic pulmonary fibrosis. So this is a chronic lung disease. It's a really high mortality rate. Again, the lungs of people with IPF have higher epigenetic ages than the lungs of controls, and there's nearly a 10-year difference in the epigenetic aging after adjusting for chronological age. This is looking at that for the Hahnemann-Horvath clocks, so it doesn't seem to be picking up in either of these clocks. So something that is really puzzling us is all these clocks are supposedly capturing the same thing. We're all trying to measure aging using DNA methylation. Why are they working differently? So why are some predicting in some things and some predicting in others? What are the characteristics of these clocks? Oh, my colors aren't showing up. That's okay. So this is the correlations after you adjust out chronological age between the clocks. So they're only correlated about 0.4 to 0.5, which means they're really capturing kind of different things. We don't know if the lack of overlap is error or if it's actually true biology. Same thing, sorry, the colors didn't show up here. This is supposed to be the CPGs that are in these different clocks. So there's only 5 CPGs shared between these 2 clocks. And most of the CPGs are unique to one of the clocks. And so far I've only been talking about these 3 clocks, but there's actually about a dozen of these different epigenetic clocks in the literature. So we're actually going through and trying to compare all 11 of these epigenetic clocks that have been published, and new ones are coming out, every few months. So we're not necessarily adding them, but one thing that we're really interested in is can you actually identify the components of the clocks and figure out which parts of the clocks are contributing to the different signals we get. So for this, we're really trying to decompose the clocks. So again, as I mentioned, there's very little overlap in which CPGs are ending up in the clocks. And this is even, for those of you familiar with genetics, we can even look at kind of larger regions. And it's not that we're just getting the CPG right next to the other one, which is a perfect proxy for it. We're getting totally different regions in these different clocks. So our hypothesis is that the reason the different clocks kind of work differently in terms of both their predictions and also what's predicting them is that they actually have different proportions of types of CPGs. So maybe different processes are being captured by these different clocks. And if you can actually break it down to these constituent parts, then we can figure out what's causing differences in this epigenetic age, why that's associated with things like mortality risk or disease risk, and from there kind of then try and put them back together. So for the aim of this, we're doing a clustering approach where we're going to cluster the CPGs into modules. So I already showed you a clustering for clustering people into groups based on their shared characteristics, but in this case, we're clustering CPGs based on their shared methylation patterns, and then we can actually compare which modules are captured in the different clocks. So again, I mentioned it's really important to consider different tissue differences. So for this, we're looking at the clustering has to be conserved across different types of tissues. So we use what's called the weighted network analysis to identify these modules that are conserved across these 6 different tissues. This is one of those dendrograms again. And what's plotted here are the age correlations, where red is a positive age correlation and blue is a negative. So what you can see is that the clocks that end up in these modules have really high positive age correlations, regardless of which tissue type you're looking at. So this is just an example of 6 different clocks. And this shows the proportion of these different modules. The modules are color-coded. And the gray essentially means these are CPGs that are not in a module. They're kind of just randomly changing methylation patterns. And what you can see is that the clocks actually have really different distributions of these different types of CPGs. And even when we get rid of the gray, for instance, the Hannum clock and our clock have extremely different patterning, or at least proportions of these types of CPGs, which is why we think that they're actually performing slightly differently in terms of their prediction. So for this, we can say, okay, what if we just take the CPGs in this turquoise module, and I recalculate each of the clocks just with those CPGs. So I set every other CPG not in that module to 0. So I can say, all right, at least the part of the turquoise in each clock, is that the same? Is that part consistent across the clocks? And basically, what I find is that for the most part, except this clock, which is a mitotic clock, you find really high consistency, at least in this signal from this. However, for this pink module, we find that maybe these 3 clocks are picking up the same signal, but these 2 are actually picking up the opposite signal. So this is actually kind of has a reverse weight in these clocks. So maybe that's what's contributing to that part. We can also look at the age correlations for the different. So this is again just the one on this side, and you can see that the age correlations are really similar regardless of which clock you're looking at, versus this one, where again you find really different patterning for this type of module. So what we're doing now. So this is really preliminary here. What we're doing now is we're actually trying to map out which of these parts are contributing to our different signals. So for instance, we're finding that maybe this cyan one in our clock is what's really driving our all-cause mortality prediction, versus the salmon one, which is the AD neuropathology associations in brains. And for both ours and the original Horvath clock, this blue module is really capturing the tissue differences. In our case, it's the opposite. We're getting tissue differences. The Horvath clock was actually trained so to have conservation across tissue. So it's actually adjusting for tissue differences. Okay, so just in conclusion, for going back to that original phenotypic age measure based on the clinical markers, basically we find that's a robust predictor of morbidity and mortality. And our models account, and collectively, when looking at what's contributing to the heterogeneity in this measure, we find that all of the factors we considered together in the HRS were contributing to about 1/3 of the variance in your difference in phenotypic and chronological age. We found that your behaviors in adulthood and also adversities in adulthood were accounting for most of this difference in phenotypic age, and that there were two groups that really had this accelerated phenotypic age, those characterized by poor neighborhood and those with that reported chronic stress. When talking about the epigenetic clock, we find that when we actually train a predictor of this phenotypic age variable rather than chronological age variable, we actually get a much better, what we consider biomarker of aging. It's more predictive of things like lifespan and health span, and also it's not disease nor tissue specific. So we're thinking it's capturing some general aging phenomenon. And then just the clock comparisons that I briefly touched on at the end. Even among all 11 clocks, there's very few CPGs or at least types of signals that are shared among the clocks. And we think that really the differences in how the clocks are performing as biomarkers are really stemming from differences in proportions of different types of CPGs. So they're capturing different aging signals. That's why some are performing better. But there is some overlap in the clocks. So even though most of the people here are probably not that interested in the biology of these CPGs, the reason I kind of wanted to show this last part was I see a lot of people who, in the social scientists want to use these epigenetic clocks, and they're going to grab, maybe the most popular one off the shelf. But what we really need to do is figuring out, based on your question, which ones are going to be best for answering that or capturing the type of biology that you're actually trying to measure. So I think, doing this kind of legwork to figure out what the different biological signals each clock is measuring or capturing is going to be really important when you then go to kind of population research and actually try and use this for epidemiology, demography, sociology questions. So finally, I just want to touch on kind of our ultimate goal is that I actually don't think an individual should have a single biological age. So again, with things like the methylation, you can get the epigenetic age of any tissue or cell in the body. And it's really, so each individual will be aging differently in each tissue or cell. So some people might be more prone to aging faster in some certain cell type or tissue type than another. And it's really your mosaic of these different aging patterns that is going to contribute to your personalized risk. We actually have to understand kind of how things are mapping on to this kind of mosaic across the entire organism. So that's the ultimate goal. We'll see if we get there. And then I just want to acknowledge my lab. My different doctoral students, my postdoc who did the HRS study with Xi Chen, my funding, and then also some, my external collaborators. And happy to take any questions.